In the extensive scientific literature surrounding Alzheimer's disease, terms like metabolic fitness and cumulative oxidative load appear constantly, but these concepts frequently operate as rhetorical placeholders. Without identifying the exact cell biological machinery that is failing, they fail to offer an actionable, targetable mechanism. To make these concepts useful, we must map them to a physical substrate. The evidence suggests this substrate is the structural integrity of the cell's internal garbage disposal, the mitochondrial and autophagy lysosomal quality control systems. Historically, research into these systems, specifically the Swerdlow program on mitochondrial cascades and the Nixon program on lysosomal failure, has developed in total isolation from the study of microglial immune collapse. This synthesis connects those isolated observations. We are constructing a testable, multi-level framework that docks 9 distinct research programs together. In this model, organelle-level failure acts as the plausible upstream driver of both neuronal plaque formation and microglial immune collapse. To understand the failure, we have to establish the baseline health of the organelle, starting with the canonical mitochondria pathway characterized by the Yule research program. When a mitochondrion loses membrane potential, PINK1 accumulates on its surface, recruiting parkin to coat the damaged membrane with ubiquitin tags. This directs an autophagosome to engulf the broken organelle before fusing with an acidic lysosome to degrade the waste. The microglial immune cell requires continuous active maintenance to sustain this metabolic state. The TREM2-mTOR signaling pathway is the specific mechanism that sustains this state, forcing the microglia to keep up its oxidative phosphorylation and glycolysis. Maintaining a healthy cellular identity relies entirely on this energetically expensive balance, detecting internal damage and destroying it before toxicity escapes into the rest of the cell. In Alzheimer's, this baseline state deteriorates. We see a simultaneous quality control failure in both neurons and microglia, starting on the neuronal side. Autolysosomes inside Alzheimer's neurons lose their acidity. Unable to dissolve contents, they swell with amyloid beta into a rosette pattern called panthos. The neuron dies from the inside out, rupturing its membrane and leaving the swollen lysosome core behind as an extracellular plaque. Meanwhile, the microglial side fails in parallel. Direct metabolic measurements of microglia exposed to amyloid beta track this precise trajectory. Oxidative phosphorylation and glycolysis spike briefly in a defensive maneuver before plummeting into a total hypometabolic flatline. This hypometabolic collapse strips the microglia of the energy it needs to execute productive phagocytic clearance. Plaque accumulation, therefore, is a coupled failure. Neurons fail to clear internal garbage, and energy-depleted microglia fail to clean up the resulting necropsy. This coupled failure generates hypotheses regarding how residual mitochondrial health dictates the ultimate fate of microglia. The first hypothesis is that the specific TGF-beta transcriptional signature required to maintain a healthy homeostatic microglia is incredibly energetically expensive to run. An age-dependent decline in this TGF-beta signaling could cause a simultaneous collapse of both the cell's genetic identity and its bioenergetic output. The cell's residual mitochondria competence acts as the sorting variable for its trajectory. High competence sustains metabolic output, allowing it to become a dam capable of productive clearance. Intermediate competence stalls the cell as an LDAM. Near-zero competence forces a purely dystrophic, dysfunctional state. The cell's internal capacity to salvage its own mitochondria gates its ultimate identity. We still have to bridge the gap between this internal organelle failure and the visible neuroinflammatory destruction observed in Alzheimer's tissue. This brings us to the single strictest convergence in this model, the Hineka research program's work on the NLRP3 inflammasome. When mitophagy fails, damaged mitochondria leak specific distress signals, reactive oxygen species, and oxidized mitochondrial DNA directly into the cellular cytoplasm. These specific mitochondrial damage signals act as the trigger, binding to receptors that initiate the assembly of the NLRP3 inflammasome complex. Once fully assembled, this molecular wheel fires highly inflammatory IL-1 beta and IL-18 cytokines outward into the surrounding brain tissue. NLRP3 is the critical amplifier, translating a private, cell-autonomous organelle failure into a tissue-destroying neuroinflammatory event. Zooming out from the cytoplasm to the neural circuit completes this three-level model. That amplified microglial effector output, gated by NLRP3, specifically targets the structural architecture of the surrounding neural tissue. The ultimate consequence is the degradation of the perineuronal nets, the structural matrices that stabilize parvalbumin-positive interneurons. Without these nets, the neurons lose the precise inhibitory timing required to coordinate the neural signals that form a memory. Cognitive decline tracks this circuit-level degradation. This suggests that the loss of memory begins with a failure in microscopic garbage disposal. Repositioning quality control failure as an upstream target changes where and when we intervene. The model generates candidate interventions, highlighting mitochondria inducers like urolithin A and NAD+ precursors. The therapeutic prediction is clear. Restoring upstream bioenergetics should preserve the protective perineuronal nets and prevent cognitive decline, even if some amyloid plaques remain in the tissue. Adding systemic metabolic interventions to current anti-amyloid monoclonals offers a way to test if substrate restoration and aggregate reduction are complementary approaches. However, the core hypotheses of this model, particularly the proposed coupling of TGF-beta signaling to mitochondrial function, remain completely untested empirically. The strength of this synthesis is that it provides a specific, falsifiable experimental roadmap designed to test these exact claims. This framework sharpens the focus from managing downstream symptoms to restoring core cellular engineering. But the hard empirical work to prove it has only just begun.